Hey everyone, it's Mr. Sinti, and today I'd like to talk to you about gene regulation. And basically, this is a discussion about how bacteria in particular are able to regulate their production of proteins. And also, we can talk a little bit about how they can affect the activity of the enzymes that are already present. And, well, why is this even necessary? Here's the question, why do bacteria need to control protein synthesis? Well, the truth is that a bacteria, if you think about it as it says here, a bacteria is sort of locked into a genome, and that is, it gets what it inherits. And so, in order to cope with changes in the environment, it can't control the future, and so it needs to exert its ability to react by affecting its own metabolic rate. In other words, speed the production of something or slow the production of something else. And so that's how it rolls. That's how it responds to changes in the environment by affecting its metabolism. And that's going to be, if you want to affect metabolism, that means you're going to need to control the rate and activity levels of enzymes. So that's what we're going to talk about. So let's jump right into that conversation. So let me make that a little bit larger so you can see. So the first thing that I want to say is that there's two basic ways in which bacteria can regulate their metabolism. Number one is by just the number of enzymes that the cell produces is one way to do it. So in other words, cells could start producing a lot of enzyme or not producing a lot of enzyme, and so that will affect obviously the rate of the reaction. So we're going to talk about how to do that. The only way to, you might predict it, how can a cell control making an enzyme? Let me just pose that question before we even start. How can a cell decide to make a protein or decide not to make a protein? You're like, well, if it needs it, it'll make it. Well, yes, but here's the thing. Like once messenger RNA leaves the nucleus, it's going to go to the ribosome and it's going to be making some protein. And so when we're talking about making a enzyme, we're talking about transcription control. So either the gene is turned on or the gene's turned off. And so it's fascinating. Two French scientists determined a little bit about how this worked, Nobel Prize winning scientists, by growing bacteria and exposing them to different medium and seeing how they react to it. And secondly, this is something that may be familiar to you. Cells can adjust their enzyme activity, in other words, how well the enzyme works, by a process called feedback inhibition. If you can recall this, in other words, how well an enzyme works. Let's talk about that. So here's something called a precursor, a chemical. And in order for the precursor to become the first product, let's just say that it's this. This is the product right here. And say this is a linear pathway leading to the production of something. Let's say that this bacteria wants to make an amino acid. This is one of the 20 amino acids, tryptophan. And so this precursor becomes A, and then this becomes B, and then lo and behold, it becomes tryptophan, which is one of the amino acids. Let's go there. And so a bacteria can make this. And it's like, it's sure you can make it, but what happens if you're grown on a petri dish like this or in a test tube? You're grown in food or agar that contains a high concentration of tryptophan. Let's do this. TYR, high concentration. It's like, don't you think it would be wasteful to keep making it over and over again if you're being given it for free? So one way in which bacteria can regulate the production of tryptophan is when it's in high concentration. This may seem familiar. Let's go try to do highlighter. The amino acid itself could feed back in the cytoplasm. Notice the negative here. It's a negative feedback. And it can actually attach allosterically, not to the active site of enzyme 1, but allosterically. In other words, if here's a picture of enzyme 1, this tryptophan can actually bond to, let's go red. So here's tryptophan in red. It can bond to a region which is distant from the active site, which will cause a three-dimensional change to the enzyme. And so it temporarily affects its shape, and therefore it slows its function down. So that's really cool. So that's temporary. So when tryptophan is in high concentration, it feeds back, screws up the shape of the enzyme temporarily, and therefore the production of A goes down, production of B goes down, production of tryptophan goes down. Therefore, you don't need to make it. It saves energy. But say you run out of tryptophan. Say the bacteria gobble it all up, and it's like, well, good thing we have a pathway to make it. And so when it's not in high concentration, obviously enzyme 1 is not allosterically inhibited, and therefore here's the pathway to produce it. So that's one way affecting the enzyme activities that are already present in the cell, especially enzyme 1 activity. Okay, that's great. Well, another way, maybe even more efficiently, that you could regulate protein synthesis is if you're given tryptophan for free in your diet. In other words, you're getting it, and you're like, yes, we just talked about this. You can feedback inhibition. Well, why do you need to make the enzyme in the first place? You don't even need to make these enzymes necessary for the production of tryptophan. Like, what would you say if there's another way? The only way to shut off the enzyme production is to stop transcription of these 5 genes necessary to produce these 3 enzymes. And you're like, how can we do that? How can we stop transcription? So you notice how part B is regulation of enzyme production. Well, just a little, here's a question. It's sort of rhetorical, but I think asking questions in the video are helpful. Then you can sort of pause and sort of think about it, like Nora the Explorer, for example. So here's my question. Here's the question. How can you block transcription? And you're like, I don't know. Well, let's tease it apart. Here's a nucleotide sequence in bacteria, and you're like, well, let's see. If RNA polymerase is the enzyme that makes RNA, and RNA polymerase attaches to the DNA, maybe if we block the attachment of RNA polymerase, then we won't be able to make no RNA, therefore no translation, no enzymes. So you guessed it. We're going to somehow need to temporarily, you don't want to permanently do this, but you want to temporarily block RNA polymerase from being able to transcribe these genes. So let's look at the detail of that. It's kind of fascinating. So the example that we like to use in most textbooks is the one that we've been talking about. It's a biosynthetic pathway that produces the amino acid tryptophan, and it involves several enzymes and many genes doing this. And so, as I mentioned before, there's 2 ways to control it. You could regulate enzyme activity, or you could regulate the number of enzymes that you're producing in the first place. And that would be gene regulation. So that's the heart of this conversation. So let's go historical here. So back in 1961, seems like a long time ago, but fairly recent in terms of biology. We have these 2 French scientists, and this is, if I were saying, Jacques, or I'm sorry, this is Jacques over here. This is Francis or Francois, I'm not sure. Francis Jacob, I'm probably mutilating it, and Jacques Manon, or Frank and Jack. Here they are right there. And they proposed that in bacteria, they were studying bacteria, and they proposed that there's a control mechanism in bacteria, very impressive, that can actually regulate gene expression. In other words, turn on the production of RNA, turn off the production of RNA. How did they figure that out? Well, they figured that inside of a bacteria, there's a group of genes called structural genes that are responsible for, let's say this is the gene of interest, making the enzymes necessary to produce tryptophan. Then there's a region upstream from the structural gene known as the promoter, and you may know this. The promoter is an area where RNA polymerase first binds to the DNA. Well, when you look at bacteria and you think there's a bunch of genes that are right next to each other, adjacent, and then there's a region where the RNA polymerase attaches. All those genes that are sort of associated with the same product, like tryptophan, that all have something, all the enzymes that have something to do with this biosynthetic pathway, they decided to call these genes an operon. In other words, all of the elements, all of the sequences of DNA that sort of work together as a team to produce a particular product. And so this is known as the tryptophan operon. It's made up of genes producing the enzyme, the promoter where RNA polymerase binds, and then there's going to be this little segment in here, and this is going to be the heart of our conversation in this video, called the operator. It's a particular small area within the promoter, still upstream from the structural genes, and it's going to be where a special protein can bond or not bond. It's kind of your on and off switch. So the operator is a place in which something called a repressor protein can either attach or deattach. So that's what we're going to be talking about, is the tryptophan operon. I can talk more about it, but there's a better picture coming. And so, like, for example, right now. So by itself, an operon can either be on or it could be off. And so let's talk about if the operon is on. So here's the tryptophan operon, and I mentioned it's composed of a promoter and the genes that are responsible for particular enzymes. In this case, it's the enzymes that make the amino acid tryptophan. So this is what it would look like if the operon is on. So the RNA polymerase attaches to the promoter. It simply moves along, doesn't start transcription until it reaches the start. And so it starts transcribing. So here's the start codon, and it moves along, reaches a stop codon, and then over here there's another gene where it's going to be transcribed, and then another gene, and then over here it reaches the termination sequence, and then hairpin loop, RNA polymerase detaches. Okay, so what's the story? Nothing new. This messenger RNA goes to the ribosome, it makes these enzymes, and there you go. So this is what happens. Bacteria make tryptophan. But remember the previous conversation. What happens if bacteria are growing on a medium that has tryptophan? And it's like, I don't want to make it. If I make it, it would be a waste. So it could regulate the production of these enzymes. And how it does it is, check this out. Focus your attention way upstream from the promoter. Notice these little lines right here mean that it's not necessarily directly next to it, but it could be further away. There's another gene, has a promoter in front of it. This promoter, notice, doesn't have an operator, so it's transcribed always. It's always being produced. So the regulator gene, it's called TRPR. So regulator gene is continuously transcribed. There's no way to control it. It sort of trickles out messenger RNA, and it makes this protein called the repressor protein. And so it's TRPR for repressor protein. But you know, it's interesting. Look at this repressor protein. They make this protein, bacteria do, in an inactive conformation, in a tertiary conformation that doesn't attach to the operator. It doesn't attach. It's inactive. And you're like, wow, that's weird. It's inactive. If it's inactive, that means that it's not going to be able to bond to the repressor. And that means the tryptophan will be synthesized. You're like, well, this is, let's see, what's going on with this? Well, if the repressor protein has tryptophan attached to it, do you notice here, tryptophan is the amino acid, so it's not drawn to scale. This is a rather huge protein. This is one amino acid. But if tryptophan attaches to a protein, it could change the three-dimensional conformation and then make it active. So check this out. Here it is, if I can get the cursor to work. Here's tryptophan attaching to a repressor protein. So in the presence of tryptophan, it makes the protein active. And if the repressor protein's active, it can bind to the operator. And if it binds to the operator, let me go back here for a second. If this right here, let me animate it. If tryptophan, if tryptophan attaches to this, it suddenly becomes active. And if it's active, it means that it's going to be able to bond to this area right here in DNA. So if the repressor protein bonds, it's sort of like a tree falling down on the track. RNA polymerase isn't going to be able to move along, And so therefore, no messenger RNA, no enzymes. So this is exactly what you want. The tryptophan, isn't this neat? The tryptophan itself is able to repress, in other words, stop. It's able to repress the production of itself. And so this is what they described as a repressible operon. In other words, tryptophan can turn off the operon. So I find that to be extremely interesting. And so it's a regulatory gene up here that produces the repressor protein. Here's a picture of the repressor protein attaching onto DNA. Cool video showing that in detail. And so tryptophan itself is considered to be a co-repressor because it's sort of like a coenzyme. It attaches to the repressor and making it active. And so normally it's weird. The repressor protein is produced inactive, but when there's tryptophan present, then it makes it active. And so tryptophan is a great example of a repressible operon. And so here's the repressor, the TRPR. It makes an inactive repressor, but then when tryptophan is present, it attaches to the promoter and then therefore blocks the transcription. And so let's go over here, and I'm going to show you this video right here I think is very good. And so this is a cool example in E. coli of an operon. Here's the 5 genes associated with the synthesis of the amino acid tryptophan right here. And so you might predict that there is a promoter, which is right over here, shown in dark blue, and that's going to be where RNA polymerase is going to attach. But do you notice here, there's a repressor protein, and if a repressor protein binds to an operator area, it's going to stop RNA polymerase from binding. And how is it able to attach? Because it has tryptophan, 2 of them attached to it. And so now it's active. But without tryptophan, it's inactive. So this is what's happening normally. In the cell produces inactive. So that means that the genes are on. And the only way to turn it off is if tryptophan is present. Because if there's no tryptophan, the bacteria need to make it. And so this is what happens. No tryptophan, RNA polymerase attaches to the promoter, cruises along, yes, it makes messenger RNA, and then that travels to the ribosome, and the ribosome makes the enzymes necessary to make tryptophan. But if there is tryptophan present, it bonds to the repressor, making it active. The repressor protein bonds to the operator sequence right there, and as you can see, RNA polymerase can't attach, and so therefore transcription is interfered with. This is an awesome diagram showing the 3-dimensional, look at the alpha helices here. This is an inactive repressor. Notice it can't attach to DNA. It's like ding, 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 ding. But if there's tryptophan present, again, not drawn to scale, 2 tryptophans can attach here and here, which then change the shape of the repressor protein, making it active, and therefore it's able to bond to the operator sequence. Therefore, DNA polymerase is not going to be able to attach to the promoter, and if it doesn't attach to a promoter, it's not going to be able to transcribe the gene. Therefore, the TRP operon is repressed. So it is a repressible operon, and who's the co-repressor? It's tryptophan. Thanks for watching. I hope you enjoyed that brief video on gene regulation in bacteria and the TRP operon. Thanks for watching.